So you need to draw the dilution method, right? Okay, so therefore there are 3 marks given. First mark is you indicate the concentration correctly, okay, and the volume that you have prepared correctly. So 20 cm of 5%, 2.5, 1.25, and 0.625. So this is 1 mark. Okay. Second mark is you indicate the W correctly. So it's 10 cm³ of W for all this. Okay, and this one here. Third mark is you indicate you transfer 10 cm³ of this solution. Okay, from one to the next one. So altogether 3 marks. And also make sure that the arrow that you draw, okay, must go into the solution, must start from the solution, must end inside the solution as well for all these 3. Okay, so altogether 3 marks. So next is the table, or there's a timing. The timing you only need to write 3 minutes. Okay, because none of the foam reaches the top in 3 minutes, so you only record 3 minutes for all. After that, you're supposed to read the height of the foam. So when you read the height of the foam, you're using ruler. Every time you use ruler, your unit must be in mm, unless stated otherwise. Just like every time you use the stopwatch, right? You write your reading in seconds, unless stated otherwise. Right? You have done 2 experiments. One experiment you write in second, right? Another one you write in minute. Okay, in minute, because they say that, okay, if it's more than 15, you write, if it's more than 15 minutes, you write 15. So that means it's telling you, please record the timing in minutes. Okay, so marking scheme. Those who just come in, please, the passcode is up there. Just make sure you mark attendance, okay? Oh, this is very small. How to make it bigger? Here. Okay, so where is my table? Top here. Table marks is heading for your independent variable is percentage concentration of enzyme, or you write concentration of enzyme stroke followed by the unit percent. Okay, then heading for your dependent variable is height of the foam stroke millimeter, and means you must have unit. Quite a number of students will not write unit 1. Okay, so you make sure you write unit. Okay, if you don't write unit, can you see you already missed out 2 marks already? It's not worth it. Okay, please remember to write unit. Record readings for all the concentration. So we don't care what are the readings, you just record. Let's say, for example, you cannot finish the exam, you just record the readings. Just write the readings, okay? That means if you cannot conduct experiment, you know the trend of the reading, please write it in full in your table, fill up the entire table. Correct trend. Correct trend means you know that when the concentration increases, right? When the concentration increases, the height of foam, which is actually the product, oxygen, right, will also increase. Right? Rate of reaction depends on enzyme concentration. So next question. So the result is 4 marks. So after that, they give you 2 unknown. Okay, 2 unknown. F1 and F2 are enzymes. So record the height of the foam in each test tube, including appropriate units. And so part 3 is between F1 and F2. F1 has a higher foam height than F2. So as long as it's higher, and your unit you must indicate. Now in this case, why they write mm or cm is because if you write cm, they already penalize you once here. So the same mistake in exam will not be penalized twice. Just the same as the same answer will not be given 2 marks in the same paper. So therefore they put mm or cm. The correct one is actually mm, but they will not penalize you again because they penalize you here already. if you make a mistake, okay? So this is for this question. The next question is you need to estimate, based on the height of the foam, you need to estimate the concentration of catalase in both F1 and F2. So this one, the examiner will look at your table, okay? Your reading for your F1 and F2, and then go to that table I've shown you just now to read off which one is the closest, okay? Which one is the closest and match with this one here, right? So you cannot write in between, you must match with any of these concentrations that's already here. So whichever that's closest, you choose that one, okay? So that's the 2 marks, all right? Correct estimation of catalase, 2 marks. Now, next question, 1A part 5. A source of error in this investigation is the difficulty of measuring the height of the foam. Now, there are a lot of possible error in practical, right? Do you know what is error or not? You know, error. Anything that doesn't allow you to get the accurate value. Okay, anything in the experiment that doesn't allow you to be able to get the accurate value that you're supposed to get for this concentration, this timing, okay, is the error. So a source of error in this investigation is the difficulty to measure the height of the foam, which is correct. Suggest an improvement to the procedure to provide a more accurate measurement of the volume of oxygen produced. So that means instead of measuring height, you can measure volume, isn't it, of the gas produced. So you need to have a special instrument. You got test tube, right? So you need to cover the test tube with a test tube bung. Okay, so this is a rubber bung with a hole that you can drill. Okay, and then you can put the capillary tube, glass capillary tube, where you can actually bend it yourself by burning it. You can design the shape by burning this side to bend it and burning this side, so it will melt. When it melts, you bend it, okay? And then you put it into maybe a measuring cylinder, okay, inside water. Okay. So you fill up the entire measuring cylinder or the gas jar with water, and then as the oxygen is released, it will push the water down. So if you use a measuring cylinder, you can measure directly from the scale of the cylinder. What is the volume of gas released? Okay, so this is only 1 mark. So look at the answer. It says, measure the volume of gas used using gas syringe or apparatus using water displacement. So this is actually water displacement or any other description, okay, that shows that this is what you meant, okay? So next question. So, okay, measure volume of gas used using apparatus using water displacement. Okay. Part 6, suggest one reason for using a 0% concentration in this investigation. So it's like enzyme catalyzed reaction. Every time you use a 0%, it means there's no enzyme, right? No enzyme means it's a control experiment. Okay, you need to understand the meaning of, do you know what is control experiment? You're conducting the same experiment, but you are changing certain factor in your experiment. Okay, I've actually mentioned to you in semester 1 that, for example, in an experiment, you have orange juice. Remember when I say orange juice, and you want to test reducing sugar as well as vitamin C. You remember that, not? Hello? Wentao remember? Cheng Han remember? Okay, because control experiment is just not to remove enzyme. It's just that those with enzyme catalyzed reaction, you remove enzyme to show what? In your experiment, when you got enzyme and substrate to give the product, enzyme has got 2 features here. Number 1 is it is a protein. Number 2, it has got active site. Okay, this is protein, and you've got active site. So you want to know whether it's protein that catalyzes a reaction or the enzyme with active site that catalyzes reaction. So therefore, when you do the control experiment, you denature the enzyme. That means to say there are two unknowns here which can affect the reaction, but I'm not sure which unknown. So I do a control experiment to find out, is it this one that catalyzes the reaction or this one itself already sufficient? So when I denature the enzyme, my active site changes shape, but my protein is still there, right? Is my amino acid still there? My protein is still there, just as it changes shape, right? So when I denature my enzyme, does it catalyze reaction? If it doesn't catalyze reaction, I have no product. Okay, no product. So it's telling me that, okay, protein alone will not catalyze reaction. It must have active site. So that means to say, when you carry experiment, there are more than one factor which can affect the rate, and you're not sure which is the factor, you have to do the control experiment. Okay, just like for example, orange juice. Okay, orange juice. I'm trying to do two tests. One is called the Benedict's test for reducing sugar. Another one is the DCPIP, which is actually the ascorbic acid test. So I got two tests, but inside orange juice has got reducing sugar. Reducing sugar, and it's got this vitamin C, ascorbic acid. Okay, so I'm not sure because I got two factors here which can affect the result of these two tests. I do not know whether vitamin C will give a positive result with Benedict's test, right? Because I learned that vitamin C will give positive test with DCPIP, isn't it? But I'm not sure whether vitamin C give a positive result for Benedict's test. I know that reducing sugar give positive for this one, but I'm not sure whether it give positive for this one. So therefore, I have to do a control experiment whereby instead of using orange juice, I use pure reducing sugar and pure vitamin C to do these two tests again. So when I do the test, if it gives me a vitamin C positive as well as positive here, you will know that when you do Benedict's test, some of the positive result comes from vitamin C, right? So that means to say that whatever color that you see doesn't really represent the total reducing sugar because some of the color come from vitamin C. So your estimation of the reducing sugar is an overestimate or underestimate. Class? Over or underestimate? Yunus? Your vitamin C so I got reducing sugar. Sorry, this is orange juice. I got reducing sugar. And I've got vitamin C. So say for example, when you test the orange juice, you get Benedict's, where's it? My Benedict is here. You get Benedict plus. Okay, brick red precipitate, a lot of precipitate, and you get your DCIP plus, whatever it means. Okay, then you're not sure whether this plus is purely due to reducing sugar or vitamin C because you got these two different factors there. Both can affect the result. So you do it separately. So for reducing sugar, you get a plus. Okay, say for example, reducing sugar, you get a plus. Your vitamin C, you also get a plus. This pure vitamin C, yeah? So their concentration has nothing to do with concentration of orange juice. It only shows that it gives you a positive result. So what can you deduce from this? These two are considered as control experiments. Okay, control experiment is not, just remove enzyme or denature enzyme. It's not. The concept of control experiment means you've got more than two factors that affect the rate. You do control experiment to find out which is the one that affects the rate. This is called control experiment. Not only I remove enzyme. No, you please learn properly the concept. So these are the, what is this one? I forgot what to say already. Control experiment. Okay, so these two are the control experiment. You do the same experiment again. So you do the two tests again. So you tell me now, based on your control experiment result, whatever you get here, is it an overestimation or underestimation of the reducing sugar? Overestimation, right? Overestimation, right? Because it could be this 3 positive comes from reducing sugar, but this positive comes from vitamin C. So this control experiment will inform you that, okay, for your balance test of orange juice, whatever color you get is an overestimation already. The actual one should be less. How less? We do not know. Okay, how less? We do not know. Same as for the DCPIP test. So I got a positive, but I do not know whether it's overestimation, underestimation, or accurate estimation. So I have to do the control experiment. So let's say reducing sugar is negative, then vitamin C is positive. Then you know that whatever estimation for vitamin C in orange juice is accurate. So this is the meaning of control experiment. Okay, so please make sure you understand the concept of control experiment here. So part 6, they remove the enzyme. So in this case here, they say why you want to remove the enzyme. So you remove enzyme to make this the control experiment because you want to find out, is it the enzyme that catalyzes the reaction? Without the enzyme, is there any foam or not? So that's why you do this experiment. So this is a control experiment to show the enzyme, to show that it is the enzyme that catalyzes the reaction. Okay, so this is part 6. Part 7, describe how you could modify this procedure to obtain more accurate estimate. So you're supposed to modify this procedure to obtain more accurate estimates of the concentration of F1 and F2. So this I have to take an example from your class. Okay. So can you tell me, Wentao, what is your F1? The foam height. 6 millimeter. F2. Okay, so this is 6 millimeter and this is 0 millimeter. So therefore, you actually need to match this 6 millimeter to here and see where it is located. So 6 millimeter is actually located here. Okay, because the unit here is actually cm. So you convert to mm, this is 10 and this is 4. So 6 is here. So that means it's nearer to 4 than 6, right? So therefore, it's actually, you have to give this reading. But you know that when you give this reading, it's not accurate. So they ask you, how are you going to get it accurate? Of course, you need to plot the graph lah. You use the data from here to plot the graph. Okay, and you draw the best fit curve. So as concentration increases, the height of foam increases. So it could be a straight line. Okay, best fit curve, straight line. So once you draw the best fit curve for the graph of height of foam, okay, versus the concentration. So this is height of foam in mm. And this is concentration in percentage, stroke percent. So then you can interpolate based on 6 mm here, you just interpolate. So for 6 mm of the height, what is the concentration? So this is the best way. You already got the results, just plot the graph and interpolate from the graph. This is the best way, okay? But there's a second way that the answer gives. The second way is, if you are not drawing the graph based on the table, you need to do something else. Now, you know that between this and this, you know that this foam height is between this concentration and this concentration, but you don't have any more reading here. So you have to bring, you have to choose this as the concentration, and you know it's not accurate. So the next thing you can do is, you want to repeat experiment again, but you use more concentration within this range. That means to say that from 1.25 to 2.5, you may want to prepare more concentration, right? For example, 1.5, okay, 2.0, okay, and then 2.25, and then all the way, so 2.25, where's my 2.25? So percent, so you may want to repeat experiment again by using more concentration within this range. And because you got more concentration now, you can actually find out for 6 mm which one it is closest to, which concentration. So this will make your concentration more accurate. You make your reading of concentration more accurate. Okay, because this one's 6 is too far from 10 and it's also far from 4. Okay, so this is the second way. So there are two methods here. Okay, either you draw the graph and interpolate from the graph of best fit curve. Must be best fit, If not best fit, cannot. Or you repeat experiment by using more concentration of catalase within this range. Then from there, read the closest concentration to the closest form of height of foam. Okay. From 6 mm, you read the closest foam height and then check what is the concentration. Okay. So this is information that you need to know how to improve the accuracy of your experiment. Okay. Next. So a data is given for you, and you need to actually plot the graph, right, in the grid given here, and they say use sharp pencil. So the entire graph, including all the writing for the graph, must be in pencil. Just like when you do biological drawing, your biological drawing and you're labeling everything in pencil. Okay. Your table, only the drawing of the lines in pencil. Your writing, all the writing in table must be in pen. Okay, so let's look at the example answer. So this is the example answer, okay? So mark is given for this. You see the X and Y axis, right? It must be labeled with units. So let's look at the mark scheme. So first marking point is the X-axis, you label it glucose concentration, stroke followed by the unit, and the Y-axis is glucose uptake rate, stroke followed by the unit. So this is format for writing the unit. You stroke it. Okay, this is mentioned in the syllabus. Okay. Then number 2, the scale they have chosen in X-axis is every 2 cm is 20 milligram. And Y-axis, every 2 cm is 1 milligram per minute. Label at least every 2 cm. That means to say that along the X-axis and Y-axis, you need to mark at least minimum 2 cm. Not minimum, maximum 2 cm. You can actually mark 1 cm. Every 1 cm, you make a mark here. But at least every 2 cm, there must be 1 mark. Okay, so don't forget to mark this one because it's mentioned. If you don't mark, you don't get the marks. Okay. And then mark all the way until the end, because some students only mark halfway. If you draw your arrow here, right, you have to mark all the way until the end. Okay, this is called systematic. Then number 3 is correct plotting of the 6 points. So the examiner will choose maybe 2 points and read whether the 2 points are accurately plotted. Okay, so remember you only can use a cross or a dot and a circle. And a cross cannot be bigger than the smaller square in the graph. Number 4 is thin, smooth line. So make sure your pencil line is thin and smooth, or else this is up to the examiner's judgment. Okay, thin, smooth line. So it's too thick. Please try to use mechanical pencil, HB. Change to HB lab. So you can use that one for drawing. Okay, from drawing biological drawing graph. Then it will always be sharp and thin. Okay, join point to point or a curve through all points. So I highly advise you to join point to point, because every time I see student draw curve, very rarely, almost all students will not get the best fit curve. Unless you got that ruler that can bend for you. So, or else usually even its best fit is actually not best fit. Okay. So best to join from point to point, because this is the fastest way for you to get 1 mark. Okay, we're not trying to achieve a beautiful curve. We're trying to achieve maximum mark. So remember that. Okay, so next question. Use your graph to suggest how glucose is transported into bacterial cells and suggest an explanation for your answer. So let's look at the graph. So the graph shows that as the concentration of glucose increases, the rate of uptake of glucose increases. So this tally with the formula for the rate of diffusion, right? Rate of diffusion is surface area times concentration gradient divided by distance. Okay, so therefore, as the concentration increases, the concentration gradient increases. So the rate of diffusion increases. So this one will tell us that the first evidence is that as concentration increases, the rate of uptake increases. So this is actually facilitated diffusion. Okay, facilitated because glucose cannot pass through the membrane directly. It has to pass through carrier protein. Okay, because it's too big. Okay, and it's hydrophobic as well. So this is the first evidence. Second evidence, you can see that it actually levels off. So this level off, it could be, okay, this level off is because your carrier proteins are saturated. Can I say equilibrium is reached? What is this? Glucose uptake rate? No. This y-axis is the rate of glucose uptake. Rate of glucose uptake. Is the rate of glucose uptake 0? No, right? It's actually 5.1, isn't it? Glucose is still being taken up at the rate of 5.1. So meaning to say that now whatever carrier protein that I have, say for example, I got 5 carrier proteins. Okay, 5 carrier proteins, all are already transporting glucose. So these are 5 carrier proteins in the membrane. 1, 2, 3, 4, 5. All are already transporting glucose. So you continue to increase glucose concentration, but you only got 5 carrier proteins. So maximum they can carry 5 per unit time, regardless of how much glucose you're going to increase in the solution. So it level off because all the carrier proteins are saturated. Okay, this thing can also happen for active transport, because active transport also use carrier protein, right? So let's say for example, these are carrier protein, but they carry out active transport. That means ATP must bind to it. So for active transport, the line will also level off, because once it reaches maximum, when all carrier proteins are saturated, you continue to add glucose, the maximum rate still remain the same. So horizontal line can also be for active transport. Okay, it can also be for active transport. So the answer is already given to you already. So you want to read the mark scheme answer just to verify. Okay, they accept facilitated diffusion or active transport. Glucose uptake is directly proportional to glucose concentration. Okay, yes. Proportional is straight line. Proportional to means when this one increases, this one also increase. So this is called proportional. So you can increase this way, you can increase this way, you can increase this way. These are all increase. Okay. Okay, so now we go to the slide question. Okay, the next question is slide question. Oh, got one more. Huh? So the question asks a student, last question for this one. Huh? So they give another table, and this is glucose uptake rate for another bacteria C. henzi. Now, if you write C. henzi in your answer, right, make sure you underline C, don't underline the dot, underline henzi. Because some students, they tend to underline the whole thing. Huh? So when you write species name, you don't underline the spacing between the two words one. So even if it's a short form also, you don't underline that spacing. And don't underline the dot. Okay. Right. So use the data in table 1.3 in your graph to suggest which population of bacteria will grow faster and suggest exponential answer. Very easy, right? Whichever that take up the glucose fastest, isn't it? So therefore, between the two, the one that take up the fastest is actually C. henzi, okay? Higher than E. coli at 40 milligram per dm cube. Okay, and then they ask you to explain why. More glucose for division is just a general statement. Okay. Because the question asks you which population of bacteria would grow the fastest. Now, every time when they say bacteria grow, bacteria, when they say bacteria grow means the bacteria divide. Okay. From one cell, it becomes two cells becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32, and so on. So the meaning of bacterial growth means increase in the number of bacteria. Of course, you say human growth, like if you grow is different thing, right? You grow in height, you grow in mass, right? Whereas for bacteria, bacterial growth means bacteria population growth means growth in number. That means you're talking about cell division. So when a cell divides, why does it need glucose? Because to be more specific, right? Because glucose is actually needed for aerobic respiration to produce ATP, which is needed for anabolic reactions, so all the reactions that synthesizes. So when a bacteria grow, when a cell divide, what do you need to synthesize? Class? What do you need to synthesize? Akila? When the cell grows and divide, what do you need to synthesize? Do you need to replicate DNA or not? So you need to synthesize DNA, right? Do you need to make new cell wall or not? Yes. Urine. Do you need to make new cell surface membrane or not? Yes. So phospholipid. Phospholipid. Do you need to make new protein or not? You need to make new protein because you need to make a lot of, which can be enzyme, isn't it? Or it can be transport protein, right? Across the membrane, right? So a lot of things need to be synthesized. So all this leads to the growth of the bacterial population. I see or not? Population of bacteria. Population bacteria grow by increasing the number, right? When you say human population increase in number. Okay, so that's why the answer is for cell division. Okay,